development, and other intellectual activities. But of course, I would like to greet Sir Ruben Berarial. Good morning, Sir. Nung ako'y totoy na instructor, ang laking tulong sa amin sa ChemEng, neighbor lang ho tayo. One time, humingi ako ng item. Sabi ni Sir, bakit? Nandito na ba yon? Sabi ko, andyan. O, go. Binigyan kagad ako ng isang item. Parang akong chairman noon, hindi ako nahihirapan humihingi. Of course, yung Dean Rico Supangco, malaking bagay ginawa sa buhay ko. Ito ay taga-pressure sa akin. Nung kami nagsimula sa biofuels noon, 2008, sabi niya mag-PhD ka. Eh, iniisip ko yung mga anak ko growing, ano? And sabi ko, hindi na ako lalabas uli, dito na lang. But after 4 years, ay nadala din. And hindi lang namalayan, nakagraduate din. So, fortunately, naging scientist pa, kasi kailangan PhD. Sir, thank you very much. Well, the rest, alam ko madami din kayong impluensya sa buhay ko and sa career ko. Yun din ang gusto kong i-share ngayon. But, you know, before itong stint ko sa Ovicre, I was going around the country. No? It all started with my boss, Dr. Hil Villancio, when we were involved sa biofuels. We went around the country. Although ako yung tinulak ng mga nakakatanda sa akin to head the alternative energy program sa UPLB. But the first legitimate biofuels program ng UPLB ay yung Jetropa, which was headed by Hil Villancio. And in fact, alam ko ay tinulungan ako ni Chancellor Berarial doon sa CHED, outstanding researcher because of the Jetropa. Di ba, sir? We were able to get some fundings and publications, no? And well, again, now when I was with my team, ang dami kong utang na loob dito sa College of Agriculture. It's very comforting. I'm very proud, no? For almost 10 years, naikot kami for feedstock development, feedstock research all over the country. Palagi kami sa Mindanao, of course, Visayas. And you will see agri graduates ng UPLB having these crucial positions sa hindi lang sa national agency, but very crucial sa amin sa baba mismo sa grassroots, no? Mga municipal agricultural officers and the provincial, and most often tutulungan kami. Sasamahan pa. Mag-ikot, mag-assess. I'm also very proud to see yung ating graduates. Every time nalabas ang College of Agriculture na board exam, ina-expect ko, ito, top na naman kami dito. Kasi medyo humihina ang ChemEng Naghahanap na lang ng inspiration. But I'm also very happy with the electrical engineering, civil, and of course the Ag-Eng. Lately, video naka-pick up kami. I'm very proud to see UPLB technology na ginagamit. One time I was asked by si Sasi pa, no? And we were in a room and they were testifying of UPLB technology. So talagang lumalaki yung tenga natin. And I was too limited ang aking exposure because being a chemical engineer, biofuels ka nga, but of course hindi mo pa alam ang feedstock. But then I was really very proud and very happy to hear that UPLB technologies ang ginagamit. And then lately, umaten-aten sa NASA, makikita mo, ang dami ng UPLB scientists. No? I'm very proud of that. And yung ating mga awards, time to time you get awards, and hindi lang sa loob, but of course sa labas. It's very comforting that ang UPLB pa rin ang nag-lead. No? Alam mo, sa buhay ko sa biofuels, which ang laking utang na loob ko, siguro kung hindi sa biofuels, hindi naman ako naging full professor before the PhD. And baka hindi din ako nagiging vice chancellor because yung track record ko palagay ko tinignan ni chancellor sa pag-pick up sa akin. And ang laking na nangyari sa akin. Ano. These are the things that I cherish. No? And I think the word today is for me is inspiration. I wanted also to share, especially sa mga young researchers, the inspiration na nangyari sa akin. And hopefully you'll be also inspired. Kung hindi man sa akin o sa Ovicre, probably dito sa college leadership and to our mentors sa college. It helped me a lot because Ako yung una, akala ko biktima ako eh. Si Dr. Art Salazar ang nagtulak sa akin when the Chancellor Ray Velasco called for a meeting in response for the enactment of biofuels law. Sabi niya, dapat meron tayong committee. And pinagtulungan ako ng dalawang vice chancellor, isa sa harap ko, isa nag-retire na, Dr. Ranyola. And tinuro ako ni Dr. Art Salazar niya, ito, gawin mong lead. Akong pinakabata doon, ano pang sasabihin mo kung ang mga senior ay ikaw nang tinuturo? I thought it was a job. But later I realized it was an opportunity that brought me where I am today. I started as a young assistant professor then, and I got the full professorship in 2014 without a PhD, all because of biofuels. And come to think of it, most of my collaboration ay dito nanggaling with College of Agriculture. It all started with, of course, partner Hil Villancio, sina Lea Villavicencio, Edwin Del Rosario, of course, si Cecil Pascual nang pumasok ng Sweet Sorghum, si Apollo Ocampo, of course, si Dean Angeles, si Dr. Rod Badayos, ang dami pa. And all of them helped me to understand yung feedstock. And later on, mas na-appreciate ko, especially when we were already working towards feasibility study and policy. No? So, ang nangyari sa akin ay malaking pagpapasalamat sa College of Agriculture. Nung ako yung nung masters ko, nagpapasalamat ako sa biotech kasi doon ako nag-thesis eh. Pero ito sa page, it was really an inspiring life, no? An inspired life working on biofuels and working with the College of Agriculture. Isang tao pa nag-push sa akin ay si Ted Mendoza. Sana wala siya rito. Pero kung marinig niya, siguro hahalikan niya ako. Kung kahit saan kami ni partner Hale Villancio, kinokontra kami niyan sa biofuels because he's not a believer of biofuels. But we are good friends, kumpare kami. On official stand niya sa biofuels, magkalaban. Pero we are good friends. And when I was given the opportunity to have a professoral chair, I proposed Jetropa. Processing of Jetropa, the waste management issue, and the economics. And then, nung ako nag-present, nandun siya, nakaupo. Lagot ako nito. Pasensya na ha, mababa ito. But I'm telling you, ito yung hindi compressed portion, hindi compressed sa full testimony. So he was there and binubugbog kami ni Partner Hill on life cycle analysis, carbon footprint, water footprint, energetics. Sabi ko, ba't alam niya to? Dapat mas alam ko to. And after the professoral chair, pumunta sa department. Sabi, Parts, yung professoral chair mo, gawin nating paper. Ko, bakit gagawang paper kalaban yung biofuels? At saka lalo na Jetropa. Tatlo sila kumontra sa amin from the College of Agriculture. And alam niyo, kinuha. And then he draft the paper. Sabi niya, finalize mo. And that was my first publication. And the rest history na. Ang dami kong publication sa practically biofuels lahat. So sa bugbog niya sa amin on life cycle analysis, especially on carbon footprint, water footprint, and energetics, yun ang naging PhD ko kasi sabi ko sa sarili ko, aralin ko to para hindi mo ko mabugbog anywhere. Inaaral ko. Nung ako'y kumpante na, I was given 3 projects, one from DOST, DA, and DOE, all on carbon footprint. Ayun ang ginamit ko for PhD. May tao na, may budget pa, and I got the PhD. Thank you for all these people who helped me from the very start. Basically, these are the people who came from the College of Agriculture, now the College of Agriculture and Food Science. So for me, it was really an inspiring time, no? When I was working on biofuels. Namimiss ko nga eh. Kaya lang, siyempre, calculated palagi yung request mo kay Chancellor. Chancellor, punta mo na ako sa project. Usapan naman namin na ikaw na bahala mag-control sa research mo, hindi kita pipigilan. But alam mo naman, ikaw ang chairman ng search committee, mga issues. So it was for me, ngayon medyo controlled na, but still hindi naman ako tinatalian. And ang problema ko ngayon, mas madami pang projects na dumadating kaysa when I was when I was not a vice chancellor, probably because of the network and because of the name of UPLB. I was imagining myself, if I'm a chemical engineer, I have this proposal, I have this idea. Kung ako hindi nanggaling sa UPLB, I don't think I will be given a chance and be approved. No? It's a different name pag narinig ang UPLB and makita ang UPLB. And I'm very happy I belong to this institution. And I'm very grateful that a numerous number of CA faculty helped me a lot in my career. So, siguro tapusin ko na, baka mamaya ay mahabang testimony. In behalf of the Office of the Vice Chancellor for Research and Extension and the whole family, we thank you. First of all, we thank you for having this symposium and hopefully tuloy-tuloy, not only during foundation, because it could help us kami sa aming trabaho, and obviously it could help us also, it could help within the college to know what other people are doing within the college. And that would open potential collaborations once you know. Ako, iniisa-isa ako din mga researchers sa atin and see kung saan makapasok. And of course, congratulations on your 109th foundation. I was able to attend yesterday, so medyo good ako kay Dean. Congratulations, because I know a lot of people in the college, the past and the present, who did a lot of contribution and exemplary dedication and service to the university and to the country. Good morning. God bless you all.
So thank you very much, sir, for a very inspiring opening remarks. So I hope that we learn and we can draw or find inspiration from all of the presentations this morning. So I'd like to call on Dr. Jocelyn D. Labias, the Associate Dean for Research and Extension and Chair of the CAFS Research Committee, to give us an overview of the symposium for today and what we are expecting in terms of the presentations.
Okay, thank you, Heidi. This must be a very exciting day. So I'm glad that I see graduate students because we encourage them to listen to the researchers of UPLB to be inspired. So today, on the second day of our symposium, we are pleased to hear from colleagues from CAFS about their research breakthroughs and developed technologies. The first is an invited paper and will be presented by Dr. Pablito Magdalita of ICROPS for his research team. We are excited to hear about the technology they developed and how it was awarded a patent only recently. Some of us here may benefit in the future about their experience in this patenting process and perhaps be provided with some tips. And equally important is how the use of their invention can benefit the food crops industry. The second paper to be presented by Dr. Jose Nestor Garcia of the Ag Systems Institute for his research team also, will detail how the commonly known gabing San Fernando that we see growing around, sometimes even unwanted, can actually be characterized to be a varying germplasm and thus can be selected for its appropriate use as food and animal feed. Their work is also a very good example of the interdisciplinary effort which we encourage here in the college. And of course, this addresses varied research questions because several units are involved, like the ASI, the NPGRL of IPB, NCPC, and of course, the Institute of Animal Science. That's a collaborative work with these researchers. Then we will hear about the 6-decade work of the College of Agriculture and Food Science Rice Varietal Improvement Group, an interdisciplinary multiple award-winning team headed by Dr. Joe Hernandez of ICROPS. They honor and sustain the work of pioneer CA rice breeder Dr. Pedro Escuro of the C463 variety, Legend. Dr. Joe Hernandez will update us on the group's recent accomplishments and plans that focus on marginalized rice areas, innovative breeding techniques, and climate-resilient varieties. Okay. Now, I remember being very concerned a few years back while driving along Laguna in my hometown province of Batangas, where we can see the hillsides and kilometers of roadsides turned almost bluish gray, with coconut trees of drooping leaves, horrified and wondering how vast the damage has been and how soon, if ever, we will recover from the coconut scale insect infestation. We could all breathe a sigh of relief that UPLB, especially the College of Agriculture and Food Science, has done its share as the go-to experts and continue to do its share now by using genomics and related technologies to render host insect resistance in some coconut varieties. This exciting breakthrough research will be presented to us today by Dr. Heidi Galvez of IPB for her interdisciplinary team of researchers from various units within CAFS and collaborators from the Philippine Coconut Authority and Boyce Thompson Institute of Ithaca, New York, USA. The last presentation for today is certainly not last in terms of importance, as it details the basic research on maintaining the post-harvest quality of dragon fruit, an introduced fruit species that is fast becoming popular. This will be presented by Mr. Angelo Castro for his team at the PHDRC. So there are 5 units represented today who will be presenting their breakthrough research, and we will all benefit from this. So ladies and gentlemen, join me in learning from and enjoying this morning's presentations. Thank you.
Thank you very much, Dr. Labios. So for this morning, like what Dr. Labios has mentioned, we have 5 presentations. We'll have the first 2, and then we'll have a health break, and then the rest of the presentations would be after the health break. We are fortunate today to have a moderator and a rapporteur for each of the sessions to help us with managing the time. I have with me the colleagues from the Institute of Weed Science, Entomology, and Plant Pathology. We have Mr. Romnick Latina and Mr. Paul Rivares to keep us with the time. And then after each presentation, we will be welcoming questions from the audience. So I hope that you brought with you a lot of questions for the discussion this morning. And before we start, we will be giving each of the presenters 20 minutes for the results and the presentation, and then we'll allot 5 to 10 minutes for the question and answer. So I hope, like I said, you find inspiration in all of these research technologies that we'll be presenting today. And we'd like to thank our Dean for being here with us, and Dr. Ben Villaria, a former Dean of the college as well. So they will be joining us in celebrating these technologies from the College of Agriculture and Food Science. So to start, our first speaker is a PhD graduate of the University of Queensland in Brisbane, Australia. At present, he is a professor of plant breeding and plant biotechnology in the Institute of Crop Science and project leader on fruit and ornamental breeding at the Institute of Plant Breeding, College of Agriculture and Food Science here at UPLB. In 2000, he was awarded as an outstanding young scientist in plant breeding by the National Academy of Science and Technology. Recently, he was promoted to be a UP Scientist 2 by the UP system. He is a prominent breeder not only on the fruits, but as you can see up front, this is one of his products. Ang ating floral arrangement. So he's very much involved in breeding ornamental plants such as the hibiscus. So let's all welcome Dr. Pablito M. Magdalita from the Institute of Plant Breeding to discuss papaya ACC oxidase gene and its use, the technology, and its patenting.
Thank you. Vice Chancellor Rex, Dean Sopangco. Of course, Dean Villarreal is here and he is wearing 2 hats at the moment. The former Dean and Chancellor of UPLB and a NAST academician. And also he's a member of our research team at IPB. And of course, Associate Dean Labios, I don't see Ma'am Tonet, but maybe she is coming. And then, of course, guests, ladies and gentlemen, good morning. My graduate students are also here. So this morning, I was tasked to present the ACC. By the way, I don't want to forget, Dr. Laurena, my partner in crime for this research, is here. So this morning, I'm going to present the ACC oxidase gene in its use and the process that it underwent for patenting. So first, let me tell you why this research was. So by virtue of Presidential Decree 729, dated June 5, 1975, IPB, under the College of Agriculture and Food Science, UPLB, was established and mandated to undertake development of new and improved varieties of important agricultural crops, germplasm collection, etc. The second one, which we are interested on, is to conduct investigations on allied disciplines related to plant breeding, such as crop biotechnology. And in 1992, under Republic Act 7308 of the Seed Industry Development Act, IPB was identified as the lead agency in crop biotechnology research in the country. And also, as mentioned by Chancellor Sanchez last Monday, this biotechnology program is in line with one of UPLB's major banner programs, hence the project on biotechnology of the ACC oxidase gene in its use. Next, please. So this morning, this is the paper, ACC oxidase gene in its use, the technology in its patenting process. I was reminded by Associate Dean Labios that they are interested on the process of patenting, how it underwent. So the paper is authored by myself and, of course, Dr. Antonio Laurena, our molecular biologist and biotechnologist, Maita Perez, another molecular biologist from the Institute of Biotechnology or Biotech, and of course, Renel Yebron from Picard, and also other people from Malaysia Agricultural Research and Development Institute. So this one is an interdisciplinary research in order to get something that is, valuable. Next, please. So the concept, what is the concept of the ACC oxidase gene? Papaya, as we all know, is a climacteric fruit, and it contains the ACC oxidase gene. It's one amino acid, cyclopropane carboxylic acid gene. And this gene is responsible for the formation of the ACC oxidase enzyme, which is the precursor of the hormone ethylene. And that hormone is the one responsible for the ripening of the papaya and other fruits, of course. Hence, the ACC oxidase gene was targeted for genetic engineering, and the purpose of that is to delay the ripening process of papaya or to extend the shelf life of papaya. Next, please. If the ACC oxidase gene is isolated from the papaya peel and then cloned in a vector or a carrier and placed in antisense or reverse orientation, there will be no ACO enzyme or ACC oxidase enzyme produced for ethylene production. And if that happens, the ripening will be delayed. The basis behind is that there will be mishybridization of the antisense mRNA, which is introduced into the plant, and the normal RNA present in the plant. So therefore, if that happens, ethylene will not be produced. If the antisense construct is then inserted into the papaya genome via, for example, agrobacterium system, the resulting transgenic papaya will produce reduced amount of ethylene, hence delaying the ripening process. Before this happens, the ACC oxidase gene must be first isolated and cloned from the donor organism, and that is the papaya. So the first thing to do is to clone the partial ACC oxidase gene of the yellow solo papaya. And to do that, good quality total RNA from fully ripe papaya samples was used in cloning the ACC oxidase gene, of course, using the reverse transcription PCR process. And as a result of that, an 800 base pairs partial cDNA sequence of the ACC oxidase gene of the yellow solo papaya was obtained. So this one is the 800 base pairs fragment that I am mentioning, which are found in lanes 2 and then lane 3. And the one in lane 4, here is the positive control, which is from the papaya of the exotica variety from Malaysia. And of course, that one is your marker. So we were confident using these primers, BFO1 and BFO2, that we got the right gene. Next, please. So it doesn't stop there. We have to sequence the ACC oxidase gene. And this one is the partial sequence of the ACC oxidase gene, which is an 834 base pairs fragment taken from the papaya. And of course, that time sequencing still has to be conducted overseas. So we sent it to the Waikato DNA sequencing facility in New Zealand so that it will be, you know, a good sequence. And this one is actually a part of the 1 kb fragment of the full ACC oxidase gene of the papaya. So that gene that I showed you has to be placed in a construct in order for it to be placed into the genome of papaya. And to do that, we use the PGA 643 plasmid, an 11.6 kb plasmid, wherein we put the antisense ACC oxidase gene here in between the SAC1 and the CEL1 restriction sites for this fragment. And upstream of that is the 35S promoter that drives this gene to work. And of course, we have the other elements, such as the NPT2, which is the selectable marker gene for transformed tissues with ACC oxidase gene, in order for it to be identified that they are the correct tissues that we transformed. And downstream, of course, is the tetracycline resistance gene, which is found in the SAC1 and the EcoR1 restriction sites to confirm for the true transformed agrobacterium. And the rest are, of course, other restriction sites. If you want to put other genes, that's okay. Next, please. So, once the gene was placed in antisense orientation, we really need to prove that it is there. So, you can find here in lanes 3 and 4, using these primers here, that the 1 kb fragment, which is this one, and the 0.8 kb fragments are all present. However, if we look at only the this one here on lane 2, we don't see this this fragment here because those ones are, you know, not containing the real antisense gene. And in lane 5, without the other restriction enzyme, you can only see this band here. In lane 6, of course, since there is no template, there is no fragment. Next, please. Also, to further confirm the presence of the ACC oxidase gene in the construct PGA01, this one is the one transformed now. So we can see that the 10.4 kb, which is found in lane 4, in the 2.4 kb fragment is present. Okay? But this one is not present in lane 3. When you digest it only with HinD3 and EcoR1 restriction enzymes. And of course, in lane 2, you cannot find any simply because this one is just the plasmid that, you know, that was not transformed. So these two fragments prove to us that we really got the antisense orientation of the genes. Next, please. So how about now the technology on transformation? So for here, we adapted the agrobacterium transformation system, and we can see in this diagram that this is the construct that we did. And of course, to remind you, the ACC oxidase gene is there, and this construct was then placed in agrobacterium tumefactions because the agrobacterium is the carrier of this gene in the construct, of course. And then we have the papaya somatic embryos that were used as the recipient of this gene. And of course, we have the agrobacterium suspension here, where the agrobacterium will be the one to put those genes inside the papaya tissue. And these are, after transformation with agrobacterium, these are the tissues that are undergoing selection for the presence, for the tissues that are truly transformed with the gene. And of course, that follows a tissue culture regeneration process. And these are the plants that we obtain, of course, which need to have a full plant because the final product of this is actually the fruit that we are interested on. Next, please. So just to show you what happened during the course of the experimentation. So these are the papaya somatic embryos. These are taken one by one out of the seeds. And these are samples of that. And there are 12,222 of those that were excised one by one out of the seed. And after that, these ones were made to proliferate into somatic embryos in Calai using the MS medium plus 2,4-D. And the percent somatic embryogenesis is from 43 to 75%. So you can see these tissues are the ones that were used. And after that, they were transformed with agrobacterium. And during the process of the selection, the kanamycin, which is a kind of antibiotic, which is coded by the NPT2 gene, and also the carbonicillin were placed into the medium. So you can now imagine the stress that you impose to the tissues. There are two kinds of antibiotics that were used. The kanamycin for the selection of the transformed tissues, and of course, the carbonicillin to kill the agrobacterium simply because you cannot make the tissues grow in the presence of the bacteria. You have to kill the bacteria as well, because if not, it will be the tissues will be overgrown. So that double stress was placed onto the tissues, but despite of that, regeneration still has to happen. So as you can see from this, these are the transformed tissues, which are brown, in contrast to the untransformed tissues, which are showing some form of greenish structures, which are regenerating into plants. Next, please. So also, we have to do a fluorescence microscopy in order to check if the cells and tissues are still alive despite of the stresses that we put onto the tissues. And to show you, these are the globular embryos that were transformed with agrobacterium. And of course, these are the still actively dividing cells that are fluorescing, just to confirm that we are in the right track of what we're doing. We cannot keep on culturing and growing something that is dead. That's why we need to confirm for the presence of that. And we have to do the fluorescence microscopy. Next, please. So the next thing is to regenerate the plant, the tissues that were transformed. And as you can see, these are the untransformed tissues. You can see the difference with the transformed tissues. The tissues that are transformed are very weak. And only this kind of tissues were obtained. And that is using, we added a brasinolide. It's a kind of brasinosoids that are taken from brassica, which is a special component of the medium that we use for regeneration. And these are the other components, the Posard medium, plus NAA, plus B, plus a benzoylaminopurine. Of course, the brasinolide. And we get this kind of tissues which are promising for regeneration into whole plants. Next, please. So after transformation, these are the kind of tissues that we get. So as you can see from panel A, a disorganized type of tissue, panel B, lip-forming tissue, and this callus with leaves, and of course, some embryo-forming tissues. And to have this is something discouraging, simply because how do we get the plants? How do we root them? Okay, so further on, after 205 days after transformation, this form this kind of structures, except for this one, which is a little bit, not really growing. But after 264 days, all these kind of tissues started forming plants like that, except for this. And these are the most promising types that we try to make some more experiments because they seem to be promising. Next, please. And at the end, this is what we got. These are the whole plant tissues, or whole plants with complete shoots and roots that we use for growing the plants. And of course, what you can see here are still torpedo-type embryos. And of course, later on, those torpedo-type embryos will regenerate into whole plants. And that is, we are now happy with what is going on. Next, please. Because transformation is like a hit and miss process, based from our former Malaysian counterparts, we were told that we need to produce at least 100 transformation events in order to get something that is useful. And we did that. And these are the samples of the tissues or the plants that we have in the laboratory. Next, please. And at the end, those plants were planted in the glass house. In the BL2 glass house, these are the plants. And of course, that's the papaya there that contains the ACC oxidase gene. But it doesn't stop there, because the next thing is we were asked to do the patenting, okay? And to do the patenting, we were happy, and we would like to acknowledge Dr. Sapangco's initiative. When he was the former director of that CTTE, he pushed me very hard to do the patenting, which I am thankful at the end. So UPLB CTTE initiated the move to patent the technology. Sabi nga sa akin ni Dr. Laurena, Pards, ikaw na ang bahala dun sa papers, because the paperwork is another thing. By the way, intellectual property rights are created to prevent others from using one's invention without one's express permission. Sabi nga po nung aming professor sa Michigan State University when I did this course on patenting, patent is quite evil, but let's see what's the importance later on. Please do not sleep. Now, the patenting process is governed in the Intellectual Property Code of the Philippines. And it's under Republic Act A293. And the rule in the Philippines is first to file. In the US, I know it's first to invent. So, pag kayo ay nagkaroon ng technology, you need to file it first. Do not divulge it to somebody or tell it, because they may get ahead of you. Next, please. So, also, another principle is that while biological organisms cannot be patented, their genes, if novel, can be patented. Hence, the papaya oxidase gene was included. Now, this one is done in coordination with CTTE regarding the patenting process, kasi po, sila ang merong expertise dito. So, first, we were asked to disclose the technology. So, full disclosure of the technology when it comes to its process and when it comes to the different publications where the technology was first put in. Next, please. So, just to give you an example, this one is the disclosure, technology disclosure form. You need to fill it up. Next, please. And the prior art search. It is very important. Why? Because the prior art search is established the novelty of the technology. It doesn't need to be new, but it needs to be novel. That's what they are looking for. And these are actually computer searches on databases of all patents on ACC oxidase genes. Like, for example, those that are deposited at the National Biotechnology Center, or NCBI in Maryland. Also, those that are at the European Patent Database. And we need to identify what is novel then with the genes that we are trying to patent. And after that, the drafting of the patent claims should be done. And it could be under the broad and narrow claims. And these broad and narrow claims should include the whereas clauses. These are the words of the lawyer. And this has to be done with a patent lawyer. For example, the broad claim will say, whereas the technology will be utilized to develop fruits and vegetables with extended shelf life and the products derived thereof. For example, the narrow claim would be like this. Whereas the technology will be used for detecting the presence of ACC oxidase gene in the final product, but not limited to papaya. And of course, all of those things has to be submitted to the patent, to the intellectual property of the Philippines office for examination. And this one is a notification of that office that a patent was submitted. And of course, we still have another one on the pipeline, the papaya ring spot virus coat protein gene. Next, please. And of course, that has to be acknowledged by the Philippine Patent Office. And that time, the fee is 1,969 pesos. Next, please. A few more slides. So the patent has to be examined by the patent office, and the applicant should quickly respond to the queries of the examiners when needed. Remember, you are talking to the lawyers this time. Of course, there should be timely provision of necessary documents like gene construct and their components with proper labels. And of course, you have to answer questions associated with previous disclosure of the technology in this case. And our experience is that we were questioned in the source of the ACC oxidase gene that Dr. Lauren and I has to justify. Next, please. So after all those things, the patent will be granted by the IPOPIL, and the award would be like this. The award grants the owners an exclusive right throughout the Philippines to make, use, sell the invention, as well as the product obtained directly or indirectly from the process. It will give you the letters patent, which was awarded in 2015, the inventor's name, the patent number. Of course, the patent life is given 20 years from the date of filing in 2008. Therefore, it will expire in 2027. So that's the letters patent that you will get after it was approved by the Philippine Patent Office. So it will be the patent application there, the date of filing in 2008, the publication in 2010, and the letters patent in 2015. Next, please. So it doesn't stop there, because we were told by CTTE that our other job is to scout for the potential users of the technology. For example, the French company called Nova Genetics in France was interested on the technology. Also, they want the PRSB-tolerant F1 hybrids. By the way, these are not GMO. And this was mentioned by Dr. Santa Cruz last Monday. Of course, we have the Hirang, the Red Plus variety, and the Timyas, which is yellow-orange. This follows the procedures for the first one, of course, is Sinta. And also, the Department of Agriculture included in their research and development and extension agenda programs for 2018 the use of the technology for our export winner crops, next please, which includes the pineapple, the banana, the queen pineapple, the mango, and of course the papaya, which ripens very, very quickly. Next please. So Dr. Lorena and I also decided that the sequence of the gene be deposited to the National Center for Biotechnology Information, a part of the U.S. National Library of Medicine, which is a branch of the National Institute of Health in Maryland, USA. And this is the transcript of the deposit there. If you click on that website, you can see this. Next, please. Also, the technology was invited to be presented in the International Association for Plant Tissue Culture and Biotechnology Congress in Orlando, Florida in 2002. And that is one of the Philippines' contribution to that big conference. Next, please. Also, it was published in the Journal of the ISAS, the cloning and characterization of partial ACC oxidase in transformation into yellow solo papaya, which was credited for my scientist. Next, please. Of course, the CTTE recognized it during the 10th year anniversary last October 17 to 18. This one is the plaque that was provided by CTTE. Next, please. And just to tell you the journey that the project underwent, to summarize. In 2001, I was trained on agrobacterium transformation in Mardi, Malaysia. And in 2002 to 2006, the application for biosafety permit to the National Committee on Biosafety of the Philippines. This has to be done because we are working on a regulated article, a regulated product. And of course, the project on gene cloning, agrobacterium transformation at UPLB was done in collaboration with Malaysia. In 2003, the paper was invited to that Congress in Florida. In 2004, there was this training on IP management, where I was sent to Michigan State University in the College of Agriculture in collaboration with the College of Law to study patenting and also drafting of patents, etc. In 2007 to 8, we did the assembly of the documents and submission to IPOPIL of the different documents. And then 2008 to 2014, this one is the long time. Examination of the patent by IPOPIL, it took a long time, no? And 2015, there was the final approval of the patent, and that is after 8 years from application. And of course, it was recognized in 2017 by CTTE, And in 2018 to 2027, that's the full commercialization of the technology. And of course, it took 14 years from experimentation to patent approval. But I hope this time will be shortened now, no? Because I hope this will not discourage people from doing patenting. Next, please. So what are the important lessons learned? I was told by Dr. Labios to share with you what are the lessons learned from this research and from the patenting that we underwent. Hindi po namin alam na ganon ang aming daranasin. But of course, like what the others, like what Dr. Rex said, when the elders are there to tell you on what to do, para pong wala ka ng choice. So let me show you the lessons learned. One, there should be the value of sincere collaboration and cooperation among team members. Dapat po, magandang good team player. Kasi po, kami ni na Dr. Laurena, kami po ay saluhan. Pag hindi ko kaya, si Dr. Laurena or si Maita Perez, hindi po kailangang magtulakan. Lagi pong nandun yung, para pong nasa volleyball, na pag hindi kaya nung isa, yun naman pong isa. And that is a very good thing that I learned. The other thing is that shuttle research fast-tracks the progress of the project. The work in Malaysia helped us a lot on the technology work and development of the transgenic. And of course, Picard was there. Capacity building of team members, dapat po talagang i-prepare ang team members, hindi lang po sa aspeto ng technical, pati po sa patenting, like what they did to me. The other one is resourcefulness of the researchers. We have to collaborate with so many people para pong ikaw ay kandidato you cannot be by your own. Yun po ang masasabi ko sa marami. No? Especially to the young ones that we are assigning to do the patenting. Of course, sabi ni Chancellor, you cannot leave or you cannot retire if you do not mentor. And that is mentoring the young and the promising researchers. Kami po may mga na-identify na, hindi pa lang po formal. Dr. Supangco will make a memo on this, he told me. And of course, nasa Diyos ang awa, nasa tao ang gawa. Okay, so those are the important lessons learned. And next, please. This is the current research team that are working with me on this. Dr. Mendoza talked to me on the revival of the genes. Please stand. So these are my research team, current research team that are working. Our motto, Dr. Villarreal is included, but he may be too sleepy this morning. Our motto is, Scientia ac labore, science and hard labor. So I cannot end up without acknowledging the people who was behind this project. One, the Picardy OST for the funding for 4 years, the ISA or the International Service for the Acquisition of Agribiotech Applications, for the IP management training in Michigan, USA, MARDI for the technical expertise in Malaysia, Biotech for a few lab apparatus, of course the CTTE for the guidance on patenting, and of course the Institute of Plant Breeding. Maraming salamat po at mabuhay.
So the floor is now open for questions. So anyone who wants to shoot the first question? Okay, ma'am.
First approval of a patent in the Philippines, because we know that there should be no patenting of life forms, right? So we cannot patent. That's why we have the PBP instead of the, right? We cannot patent life forms. But I do not know because genes are part of the biological system, and I really do not know. It can be challenged.
Actually, it was accepted and patented as what we all us can see. Because genes, if novel, can be patented. I think Dr. Laurena can tell you more of that.
There's a new ruling now that under the World Patent Office, you cannot patent genes. But in our case, we patented the cDNA, and the cDNA of the gene can be patented.
So the gene and
only the cDNA. Since we use a reverse transcription PCR, then we focus on the cDNA.
Okay, that's clear now. So you cannot patent the gene because the gene is part of the system, right?
Formerly it can be patented.
Now
Anyone else?
The presentation was very informative and it is very attuned to the needs of time. My question is, okay, sir, the patent lasts for 20 years. My question is, what happens to the intellectual property rights of the inventor after the expiration of the patent?
Well, it finishes on that, but if you really want to extend, so you may renew. But then 20 years, of course, is already long, so that the use will be, available for the masses and the public. May gusto pong sabihin si Dr. Supangco.
You can renew, but if you renew, there must be some additional new improvements to the technology. Otherwise, if it's the same thing, identical, it will not be allowed to be renewed. It becomes public knowledge. I have a question, which is actually out of the box. This is what President Concepcion usually wants. If you know that gene, and then how it affects ripening, is it possible to create something outside of the plant using that gene, that when you spray it to the fruits, it would do the same thing?
Actually, the post-harvest people are doing that because I think one that they are using at the moment is MCP. So that one is an exogenous application of some chemicals also to delay the ripening, and of course some anti-ethylene compounds, like for example silver nitrate, and also the other one is your silver nitrate, and of course the other one is sodium thiosulfate. So those are ethylene scrubbers that would also delay the ripening. And also I would like to add that the genes are not the only ones that are patented. Remember, it's the ACC oxidase gene in its use that was patented. That's the one that was applied for. It's not only the cDNA that was patented. It was also the process and the products that are derived from it, as what we have put up in the document.
Okay. So we'll be entertaining just last one question for this session.
I don't think this is a question, but rather to reiterate the conclusion of Dr. Magdalita that it is very important to have teamwork. Teamwork is very important. Hindi kaya ng isang lahat nagampanan ang anumang ori ng suliranin. Subalit kapag tulong-tulong, yan magagawa ng paraan at magkakaroon ng solusyon. At yung kakalimutan din, nasa Diyos ang awa at nasa tao ang gawa. Thank you.
Thank you, sir, for your words of wisdom. Thank you, sir Magdalita, for your wonderful talk. Let's give him a round of applause once again.
So we'd like to call on Vice Chancellor Rex for just a minute, and then our Dean Supangco and Dr. Villarreal to assist in giving our certificate of appreciation and the small token for our speaker for today. So let me read the citation. This certificate of appreciation is presented to Dr. Pablito M. Magdalita for serving as resource person on the topic Papaya ACC oxidase gene and its use, the technology and its patenting, held this 8th day of March 2018 during the R&D Symposium with the theme Research, Innovation, Development, and Entrepreneurship Towards Inclusive Growth, held at the Post-Harvest Horticulture Training and Research Center Annex Building, College of Agriculture and Food Science, UPLB, signed Associate Dean for Research and Extension, Dr. Jocelyn D. Labios, and Dr. Enrico Piso Panco, our Dean of the College of Agriculture and Food Science. So thank you very much for a very informative presentation for this morning.
Okay, so now we'll proceed with our next presenter. He is the head of the Division of the Agricultural Systems Studies of the Agricultural Systems Institute. He is the program coordinator of the Adaptation to Climate Change program of the ASI and the program leader of the EIDR-funded project Interdisciplinary Research and Development for the improvement of the production and utilization of Gabing San Fernando for Gabing San Fernando for feeds. Let us all welcome Dr. Jose Nestor M. Garcia.
Good morning, everyone. Happy anniversary, College of Agriculture and Food Science. Okay, so the program that I'll be presenting is just the highlights now. The program that operationalize interdisciplinary research and development undertakings to optimize the benefits from Gabing San Fernando. Kilala ba natin itong Gabing San Fernando? No? Santo Soma Sagitipolyo. So, iba to sa taro, no? This is different from taro. Okay, so we're doing this through identification of suitable germplasm. This involves the production and utilization continuum of the crop and development of production technologies and utilization of this crop as feed to swine. No? Okay. Uy, ba't ganun? Mali ata. Yeah, okay. So, ba't kailan natin start pag-aralan itong Gabing San Fernando? No? So, Gabing San Fernando is considered as alternative source of food, feed, and fuel. So, malaki ang papel niya at malaking potential niya. No? Lalo na ngayon meron tayong climate change. And yung production technologies natin sa Gabing San Fernando ay wala pa, hindi pa na-establish natin sa country natin, no? Sa bansa. At ito ay based on farmers' experiences lang, kalimitan. No, wala tayong technology dito. So it remains to be an understudied crop in the country and very limited scientific research na kinundak dito. No? So nag-search kami sa buong wala tayong masyadong research about this. No? Marami sa ibang bansa, pero sa atin wala pa. No? So there's a need to develop science-based component technologies for large-scale planting of this crop. Wala naman. Pabalik tayo yung... Ayan. Okay? Sige ka na lang, sige. So, ang objective ng program na to ay includes the following. No? Identify suitable germplasm as feed to swine. Ang feed nito ay, we're utilizing the corm. No? Yung corm, yung parang trunk niya. And the cormels are the ones being marketed, being used as food. Yung corm niya, kalimitan, tinatapon na lang after harvest ng cormel. So we are utilizing that corm for feed to swine. No? And develop component technologies for Gabing San Fernando, which is ngayon wala pa masyado. And develop high-quality swine feed products and also compile publications on Gabing San Fernando germplasm, production, and utilization. No? Next. Okay, so the program consists of 3 projects. Yung unang project ay ito yung collection, characterization, and evaluation of Gabing San Fernando germplasm. No? This involves passport data, morphological and agronomic characterization of yung mga collections natin, then approximate chemical composition, nutrient, and anti-nutrient, no? ang gabing San Fernando ay maraming oxalate, mataas ang oxalate niya, no? Ito yung nagkukos ng kati, no? So that's an anti-nutrient. Then we are also cataloging yung mga collections natin ng gabing San Fernando. Then the second project is the development of component technology in farming systems, no? So this involves propagation techniques for rapid multiplication. Then productive planting cooperation, ano ba yung distance of planting niya? Then soil fertility management and development of integrated pest management for the crop, no? And the third project is on the development of feed products from gabing San Fernando. So this includes chemical and nutritional characterization of different byproducts of gabing San Fernando and developing processing techniques to reduce anti-nutrient, no, yung oxalate. Then testing this gabing San Fernando-based feeds sa performance dun sa baboy. Next, please. So, as to the highlights of our findings, so we were able to collect a total of 111 gabing San Fernando accession in 41 provinces throughout the country. That is in addition to the 99 collections already with the NPGR. And we were able to conserve and generated a total of 241 accessions, and 17 of which were already duplicated in tissue culture laboratory, no, sa IPB. And we were able to conduct diversity and cluster analysis for 149 accessions na nakolek namin, and conducted oxalate and proximate analysis for the accessions. And from that, we were able to select promising accessions para doon sa feeds. Next, please. Okay, so based on our analysis, we were able to observe high diversity of 5 qualitative characteristics ng accession, no? Then medium diversity on 7 characteristics and low on 10 characteristics, okay? And for qualitative characteristics, we were able to observe high diversity dun sa 10 characteristics, then medium on 5 characteristics, and low on 2 characteristics. Yan yung nasa table na nakapresent dyan. Okay? Next, please. So we were able to characterize also morphologically and photo-documented 42 accessions, and determine proximate analysis and oxalate content, no? And from that, we selected 12 promising accession based on morphological traits and low oxalate content, no? Then we were able to catalog yung mga collections namin. So, as to the development of component technologies, we found out that the use of whole corm, no? yung trunk niya, is the most effective propagation material because it will sprout early. It gives highest percentage sprouting and high corm yields, although no significant observed with other treatments on corm yield. Corm meal ito, no? It's not the corm. No? Pero in terms of the corm, maganda siya. And the use of set, yung nasa picture, this is a set, no? It's also effective regardless of the size of the corm set and length of petiole, yung length ng petiole niya. Ito, no? But retaining the shoots, yung nakita namin, pag i-retain mo yung shoots niya with the set, would reduce percent sprouting, no? So kailangan putulin din yung shoot. And generally, small corm with short or long sets significantly give highest corm yield, and small corm set with 15 centimeter petiole and big corm set with 30 centimeter petiole gives the highest cormel yields, no? Cormel naman ito. And using the corm's top half, yung top, yung taas niya, no? And its quarter and small cormels, yung mga hindi na namamarket, yung mga palingin tinatapon na, can also be recommended in the multiplication of propagules. Okay, next please. And we found out that gabing San Fernando performs better under shaded condition, no? Rather than in open field. Although sa wild may makikita tayong mga sa open, mga gabing San Fernando, pero generally, mas maganda sa shaded condition. At yung in terms of spacing, ang nakita naming maganda ay itong one by one spacing, no? because it performed the best sa lahat ng ano. Although sa yield naman ay wala masyadong difference. And we found out that the application of vermicompost and mycobam gave yields comparable with the application of inorganic fertilizer, yung 60 kilograms NPK per hectare. No? Nandito yan sa ano. Okay, in terms of yung pest naman, we were able to observe 14 insect pests, no? And diseases, and 8 beneficial insects doon sa Gabing San Fernando. No? And the presence of banana aphids may indicate the possibility of the insect to be carrier of Dacian mosaic virus ng Gabing San Fernando. So there's a need to seriously consider the emerging threat of rhinoceros beetle. No? Nakita namin yung rhinoceros beetle doon sa Gabing San Fernando on areas with coconut and other palms kasi doon sila marami. No? Marami doon ang rhinoceros beetle because of the possible severe damage on corm and cormel quality. Okay, next, please. So, for the development of feed product from Gabing San Fernando, dahil nga sabi ko kanina, mataas ang oxalate content ng Gabing San Fernando, but we observed that yung calcium oxalate ng Gabing San Fernando increases with age nung corm niya, but processing treatments such as boiling and fermentation significantly reduce its level. No? And the approximate calcium and phosphorus analysis of the basal diets conform closely to the corresponding calculated nutrient analysis. Next, please. Okay, then we also developed some Gabing San Fernando-based diets and tested it with for swine and compare it with corn-based diets, no? Yung mga usual ano natin. So we're trying to substitute yung corn ingredient of the feeds with Gabing San Fernando, no? And we found out that yung mga Gabing San Fernando-based diets give comparable performance sa pig, no? Comparable with the control, yung corn-based. In terms of average daily gain, feed efficiency, caloric efficiency, dressing percentage, and back fat thickness. And in the growing phase, pigs fed with Gabing San Fernando-based diets have lower feed intake, no? Compared dun sa corn-based. Mas konti yung nakakain niya, but it's not affecting the body weight. No? So magandang ano ito. And in the finisher phase, pigs fed with fermented Gabing San Fernando corn diet had the lowest feed intake. Probably because of the high energy content of the feeds. Kasi dun sa fermentation, we added molasses, no? Sa fermentation process. And feed cost efficiency of both grower and finisher diets was not statistically significant among treatments. No? So, ibig sabihin, comparable lang sila dun sa, in terms of cost. Okay? So, for future plans, we were on the process of publishing yung catalog natin on Santos Soma Collections. We have a total of 56 dun sa catalog natin. And we will finish yung compendium natin on Gabing San Fernando, which is the first in the Philippines. Wala pang ganitong nakita namin sa Philippines. And publish articles in scientific journals. We targeted 3 at nakapag-publish na kami ng isa. And we will write yung production manual ng Gabing San Fernando. No? And also prepare proposals as a follow-up sa program na to on establish the economics of commercial feed production using Gabing San Fernando. No? And also Gabing San Fernando-based food product development. Kanina feed yun, tapos food, siguro in collaboration with food science siguro. Okay? Next, please. So, ito yung Santos Soma team or ex-team. No? So, makita nyo yung mga members niyan ay coming from different units of the College of Agriculture and Food Science. No? And most of them are here, ready to answer your questions. Thank you very much.
Thank you, sir, for sharing with us your research on Gabing San Fernando. So the floor is now open for your questions. Anyone? Sir.
Nestor, congratulations. I thought that this is a very good study. I remember when this was still being reviewed, I was one of the reviewers, and I'm glad that it is now being implemented. My question, Nestor, has something to do with the comparable composition of Gabing San Fernando, of corn and commercial formulations. In other words, you said that they have comparable performance, but I wonder when you compare, how would the composition of Gabing San Fernando compared with those of corn and the commercial feeds?
It's only the core components are that we replace with Gabing San Fernando. So it's a substitution of core ingredient with Gabing San Fernando. The rest are all the same.
And nevertheless, okay. So it's the same. Mm-hmm. Thank you.
Anyone else?
Sir, I have a question on one of your component projects or technologies that you'd like to develop was actually on pest management. So have you established some baseline data on, let's say, impact of this insect and also the diseases? And I'm really interested in the weeds part, sir. So meron po ba tayong data on that with regards to yield?
So, kung sa pest naman, pag-usapan natin, nag-ikot kami sa maraming parte sa Pilipinas and we observed the pest. No? So, nakikita namin yung 14 pest. Pero, hindi sila masyadong threat na doon sa gamit sa Fernando. So, wala masyadong damage. No? Wala masyadong, konti lang yung mga...
Okay, so I saw another hand here. Yes, ma'am.
In feed formulation, the most important component is actually percent protein, right? So how does Gabing San Fernando compared with the corn-based formulation?
So, medyo comparable siya. We have
in terms of protein content?
Okay.
Yeah.
Yes. Meron tayong 4.75 content ng natin, ng Gabing San Fernando. No?
Ang corn, di ba, mga 7?
Si Marty ang may sagot niya.
Ako po kasi yung parang nag-task. Actually, we have a study na comparison sa cassava because the cassava is the alternate to corn kapag mataas yung price. So we wanted to maybe compete with cassava rather than corn because it's a complete, walang supplementation or whatever. But we found out on the collection that we have a cassava accession with 6% protein. I'm sorry. Gabing San Fernando with 6% protein. And generally, according to Food and Agriculture Organization as well, Gabing San Fernando has the highest protein content in terms of the tubers. Like, it is comparable with potato and mas mataas pa nga pa siya sa yam. So, and in terms of essential amino acids, like in cassava, Gabing San Fernando has 5 times more lysine and twice as much methionine compared to cassava. So, ang problema kasi ng cassava, nagsusupplement tayo ng essential amino acids sa feeds. So, maybe because we have an accession na mataas yung protein content, baka hindi na tayo kailangan mag-supplement ng essential amino acids because it's higher.
My question is this. Gabi San Fernando is actually produced for its corms, right? So, cormels. So, what you're using is a waste product. Yeah. So, I'm just thinking that when you analyze your cost and return, you should also include income from the main product, which is cormels. Yeah? Kasi itong na to is basically using the waste material for another component of your farming system, which is swine. So when you analyze this, you should put this in the context of the whole farming system rather than just individual, because it might not provide you with a very clear advantage when you talk about just the Gabi San Fernando for swine feeding.
So, yung core meal, which is the one being marketed, yung binibenta, ay hindi namin ginagalaw yun. Although we consider that in the selection of the germplasm. No, doon sa selection, tinitingnan din natin yung core yield. So, dapat mataas ang corn yield, at mataas ang protein.
Because anyway, because it all depends on your breeding objective. If your breeding objective is really towards for animal feed, then you select more on towards that, the yield of the corn. But if your objective is towards the marketable, then you would more select towards that, and then look at the waste product and put it to the livestock. So I think that should be clear. What is your really objective? Is it towards the marketable crops or towards animal feeds? Because it's very difficult to put them together at the same time, because one would affect the other.
So, pinaka-objective talaga yung mataas ang core meal, which is being marketed, pero mas maganda rin kung mataas din yung core meal. So, double ano.
So we'll be entertaining just the last one question. Anyone?
Good morning. I would like to ask how extensive is the San Fernando Gabi production if you are proposing it to be a substitute for cassava as feed or for corn, how extensive is it? And how would you propose to disseminate all these things? Saan ba ang mga production areas?
So, actually, dahil nga hindi masyadong pinapansin itong gabing San Fernando, no? But you can observe this in the wild, no? Ang dami-dami. Pag magbiyahe ka from here going to Bitin, ang dami sa gilid-gilid doon. Actually, doon kami nakakuha ng mga planting materials, no? So, they're just in the wild growing, no? So, kung ano natin sa mga farmers na ipaano natin sa kanila na kikita sila dito, not only from the core meal, but also from the corn as feeds to livestock. So, may inganyo sila, no? Actually, itong gabing San Fernando ay this is being used already as feeds sa native swine doon sa Mulanay, Quezon. Mayroon kami project doon. But they're cooking the corn, no? They're selling the corn meals, but they're cooking the corn as feed to the animals. That's why we're trying to develop yung mga ibang process para ano. Pero they're planting already gabing San Fernando under coconut, no? Which is a shaded area, and sabi natin is maganda yung performance niya under shaded areas. So ito talaga promotion talaga, no? Alam natin sa mga magsasaka na kikita sa dito, no? Sa gabing San Fernando. Pero there are a lot of areas na pwedeng matamnan ng gabing San Fernando. Ang potential niyan under coconut, so malaking area natin ng coconut na walang intercrops, no? Sa ilalim. So we can promote in those areas. And also, so wild nga, marami nga, no? Yung mga tabi-tabi lang, pwedeng itanim doon yung gabing San Fernando. Okay?
Okay, thank you, Sir Nestor. So, guys, should you have any concerns about the studies they presented, the both presenters, you can always, I think they're very much accommodating to entertain your questions personally. Okay, so for today, we end our first session by giving certificates to Dr. Nestor Garcia.
Okay, so let me read the citation. The certificate of appreciation is presented to Dr. Jose Nestor M. Garcia for serving as a resource person on the topic germplasm characterization, production, and utilization of gabing San Fernando for animal feeds, held the 7th day of March 2018 during the R&D Symposium with the theme Research, Innovation, Development, and Entrepreneurship Towards Inclusive Growth, held at the Post-Harvest Horticulture Training Research Center Annex Building, CAFS UPLB. Signed, Dr. Jocelyn D. Labios, Associate Dean for Research and Extension, and Dr. Enrico P. Sopanco, the Dean of the College. So we will have a break, and then be back here at 10:15 for our second session for this morning.